Alright, so this is where it all comes together. We start to look at how the muscles do their job. It seems very simple. Stimulate a muscle because it's excitable, it contracts, stop stimulating it, and it relaxes. Alright, but how exactly does that occur? And it gets down to, as you would imagine, down to the cellular level, and as we were just introducing, down to the molecular level. We're talking about individual proteins that are interacting inside of the muscle cell and inside of the myofibril, right? Like I said, layers upon layers upon layers upon layers. So, what we're going to look at are the steps involved in this contraction process. So this is a key thing. So two aspects about this, right? Number one, like we said, we have to understand the anatomy and then the physiology. We've laid out the anatomy in terms of the different parts. Now we have to talk about how those parts work. So the other thing about it is this is our good first step as we're getting into our different, more complex body systems, where we're actually having to understand, like we mentioned, the very small level intricacies of how the body functions. So you got to kind of get used to that. So one of the major topics then is going to be the sliding filament mechanism. The other one is going to be talking about what happens at the neuromuscular junction. When the neuron is stimulating the muscle cell, how does that signal get across, and how is that transmitted from one system to another? So as we get to nervous system, another excitable system, we'll have to talk about how these cells, also those cells, will basically respond when they get stimulated. All right, but that's for the future. All right, so what we're going to be looking at is, again, the contraction. Now, we talked about activation of crossbridges. Now, if you remember when we were looking at the structure of the myofibrils, we actually had our individual myosin molecules that look kind of like golf clubs, and we had here an area that was called the head, right? So like the golf club head. So these are also called cross bridges, because what they're going to do is they're going to make bridges. We're going to make bridges with the actin filaments. So these are going to come out like this, and they're going to come across, and they're going to make an attachment temporarily to the actin filament, and then they're going to do a little pivoting or ratcheting action. Okay? So that's where we see the term cross bridge come up. Okay, so let's talk about how this is going to occur. So in terms of this model of contraction, which took some time to actually discover, right? Once upon a time, we just thought there was like little mice running around underneath there. It wasn't until we had the technology to look inside of cells and down at this molecular level to actually figure out what was going on and how these different proteins were interacting. All right, so here's the key aspect, though. We have those thick filaments, right, the myosin filaments, and we have the thin filaments. And what they're going to be doing is they are going to be sliding past one another, okay? So it's going to be kind of connecting, it's going to be ratcheting, it's going to be pushing that thin filament past the thick filaments. And that is going to cause that sarcomere, the functional unit of the muscle, it's going to cause that sarcomere to contract. Right? The thin filaments and those Z lines, like where my hands are, they're going to be pulled and pulled and pulled, like pulling on a tug-of-war rope. And as they come closer together, then we're going to reach this state where the sarcomere is fully contracted. And the sarcomeres make up the myofibrils, which make up the cell, which make up the fascicle, which make up the muscle. And so if all those smaller things are getting shorter, then the muscle itself is getting shorter. Okay? But what we're going to find is that the thick and thin filaments are sliding past one another, right? The thick and thin filaments themselves are not changing length. They're just sliding past one another, kind of like this, okay? My individual fingers are not getting any smaller, but the palms of my hands are getting closer together as my fingers slide past one another, okay? Okay, so let's remind ourselves of the players here, right? So we're going to see referrals to A band and I band and Z disc and that type of thing. So we're going to take a look at what's going on there. So remind ourselves, so here's our thick filament in pink, thin filaments in blue like you see here, Z lines to the sides. A band is where we have overlapping thick and thin filaments and they appear dark, and an I band is where we have just thin filaments. So it's going to be all across here like you see. So here's our I band. That's the border between our I band and our A band. I band and A band. and Z line like we see there. Okay? Okay, so notice difference between relaxed and contracted. So see how wide our sarcomere is here? From here, way over to here. Okay? And notice that H zone. The H zone in the center is where we just have thick filaments and no thin filaments. But as those thin filaments are being pulled in, that H zone is going to get encroached upon, and smaller until it basically disappears. And that's what we see here, right? So we see the H zone basically disappearing. We don't have any thick filaments just by themselves, okay? And when we look over here, even the I band, like we see here, is getting smaller because an area where we have just thin filaments, the thick filaments are being pulled, the thin filaments are being pulled, and the thick filaments are kind of filling in that gap as well. Now the A band, the width of the A band itself doesn't change because the width, like we said, of these different fibers, the width of these fibers don't change. And the A band is defined as like from the one length end of the thick filament to the other end of the thick filament. And the sliding filaments, the thin filaments are sliding past it, but that width of the thick filament isn't going to get any shorter. Okay, so the A band doesn't change. Okay, so like we said, two major topics in here. One is, how do we get that muscle to contract? This is voluntary, but we have to have the nervous system telling it when to do so. So we need to understand that process. And then once the signal gets to the muscle cell, how does it respond? So we have 4 steps to stimulate a skeletal muscle cell, right? So we have the nervous stimulation, we have the generation of an action potential, and then the action potential moves along the sarcolemma. Now what's a sarcolemma? Dora the Explorer moment. Plasma membrane, right? So it's going to be propagated along the plasma membrane, but then what it has to do is that signal has to get down into the cell, right? So we have those T tubules that are going to go down in, so we have to get in amongst those myofibrils, and then this flood of calcium is going to occur coming from the sarcoplasmic reticulum, okay? So steps 1 and 2 here occur at what's called the neuromuscular junction, neuromuscular, and then steps 3 and 4 are occurring in the muscle itself. All right, so that's what we see, kind of our different phases that we're going to see here, as well in terms of getting this muscle fiber to contract. All right, so we said that where the stimulation is going to occur is at the neuromuscular junction. It's also called the motor end plate. Now we haven't talked a lot about our different, we haven't gotten to nervous system yet, but we have done a tour of all of our different cells in the body, right? So we looked at the neurons. Neurons are kind of star-shaped cells and usually have one long cellular extension. It's called the axon, and that's where the signal is going to be sent down or propagated in order to get to the next cell. Now that next cell could be another neuron, it could be a gland, or in this case it could be a muscle, okay? So we're going to see the neuron come down with its axon down to where the muscle is, and then it's going to branch, right? And then we're going to have these little motor end plates where the interaction is going to occur. And that's what we see here. So here's the neuron coming down from the top right here. Okay? So this is a separate nerve cell. That's the axon coming down. The action potential, the signal is coming down. And then this is the place here with that little gecko foot like you see there, right? The axon terminal. That's the motor end plate where we're going to interact with the muscle cell itself. So here's our muscle cell, multinucleate with the striations. We're going to zoom up on that. So when we look at that little expansive area of the axon terminal where it looks like almost like a gecko's toe, okay, where it expands like that, this is called the synaptic end bulb, right? It's at the end. And so down here, what's going to happen is the action potential is going to arrive at this last little part of the axon, and inside of it, there's lots of different things. It's part of a cell, right? So we can see mitochondria there. You can see a variety of different things. But you'll also notice that there are these little ball-like structures here, okay? These are vesicles. These are bags, membrane-bound bags, that have neurotransmitter in them. It's synthesized up in the body, in the main star-shaped area of the neuron, but it's transported down to this area because these bags of neurotransmitters, these are the chemical messenger that's going to go and stimulate the muscle cell. So that's where they need to be. So once that action potential gets down there, notice right here, sorry that it's kind of small, but right there, says Ca2. That's calcium. So calcium channels, these are electrically gated calcium channels, open up, right? They're getting a bzz, bzz stimulus to tell them to open up. And when they open up, calcium diffuses from the outside of the cell, the extracellular environment, to the inside of the cell because of a concentration gradient. Remember, it goes downhill. So if it's high outside the cell and it's low inside the cell, when we open that gate, it's going to diffuse into the cell itself, into that neuron. And that diffusion of calcium basically is the signal that tells these vesicles to go down here and to merge with the plasma membrane of the neuron and to dump out and release its neurotransmitter. And it releases its neurotransmitter here into this gap. Okay? All right, so like we said, voltage-gated calcium channels are going to open up. Calcium is going to diffuse into the cell, and it's going to stimulate our different synaptic vesicles. Now notice here, it says ACH, achoo, kind of. But ACH is acetylcholine. That's the specific neurotransmitter that is used at the neuromuscular junction for skeletal muscles. There's a lot of other neurotransmitters, but this is the specific one. So when I say neurotransmitter, that's acetylcholine, okay? When I say neurotransmitter, you say ACH. Neurotransmitter. Exactly. At least at the neuromuscular junction. Okay, so the acetylcholine is released and it moves out via excitosis. Exocytosis is going to release it into the gap. Now we have a concentration gradient for the neurotransmitter, right? It's high where it's just opened up and it's low on the other side of the gap. So it also diffuses. It goes across the gap and it binds to receptors that are found on the motor end plate. We can see them right here. So see these here? These are the receptors on the muscle itself, right? This is the muscle fiber down here. So those are receptors that are on the muscle fiber, and they're called chemically gated channels or ligand-gated channels. So what gets them to open up is the arrival of that chemical messenger. And once they bind it, then they open up, okay? And that's going to lead to the stimulation of the muscle cell itself, okay? So we see ACH, acetylcholine, that's the neurotransmitter. And so that's going to diffuse across the synaptic cleft, and it's going to bind to receptors on the other side. So notice, right? Notice this blue area right here? That's called the cleft. That's the gap. Physically, while the neuron and its axon terminals are right near the muscle cell, there's always a space between them. So how are we going to get a message across that space? It's kind of like if, today it doesn't much matter, but like when I was a kid, before we had cell phones and stuff like that, if you wanted to communicate with somebody across the room, you might write it on a note and you might fling it across the room because there's a gap between you and who you want to talk to, and that gets your message across. So that's the way that the neurotransmitter is also getting a message from the neuron to the muscle cell itself. Binds to a chemically gated channel. Okay, the channel opens. Now we're actually going to have a flood of sodium going into the channel, causing an action potential, a rapid change in membrane potential in that muscle cell. So that's where we can see down the bottom there where it's going to be binding to those receptors in the motor end plate itself. So you can see how it binds right on the channel itself. Okay, so we've got the signal across. So now the muscle cell is receiving the signal. But then the question is, okay, we've stimulated it. It's going to contract. We're going to talk about how it's going to contract. But then the question is, how do we stop stimulating it? We just rain down a bunch of neurotransmitter on it. How do we get rid of that? So what we find is that there's an enzyme, look, ASE. If you find a term, not all of them, but most of them, where it's a molecule and it ends in ASE as a suffix, that means it's an enzyme, which is a catalyst, which, remember, catalysts or enzymes are going to facilitate chemical reactions, make these chemical reactions occur a little bit easier. So acetylcholinesterase is an enzyme that helps to break down the neurotransmitter. And once we break apart that chemical messenger, now the message isn't getting through anymore, so now the muscle can actually relax. All right, let me see if I can go and play a video for us.
Nerve impulses, also known as action potentials, travel from the brain or spinal cord to trigger the contraction of skeletal muscles. An action potential propagates down a motor neuron to a skeletal muscle fiber. The site where a motor neuron excites a skeletal muscle fiber is called the neuromuscular junction. This junction is a chemical synapse consisting of the points of contact between the axon terminals of a motor neuron and the motor end plate of a skeletal muscle fiber. The events at the neuromuscular junction occur in 7 coordinated steps. Step 1. An action potential travels the length of the axon of a motor neuron to an axon terminal. Step 2. Voltage-gated calcium channels open and calcium ions diffuse into the terminal. Step 3. Calcium entry causes synaptic vesicles to release acetylcholine via exocytosis. Step 4. Acetylcholine diffuses across the synaptic cleft and binds to acetylcholine receptors, which contain ligand-gated cation channels. Step 5. These ligand-gated cation channels open. Step 6. Sodium ions, shown here in red, enter the muscle fiber, and potassium ions, shown here in blue, exit the muscle fiber. The greater inward flux of sodium ions relative to the outward flux of potassium ions causes the membrane potential to become less negative. Step 7. Once the membrane potential reaches a threshold value, an action potential propagates along the sarcolemma.
You can see those T-tubules going down inside.
Neural transmission to a muscle fiber ceases when acetylcholine is removed from the synaptic cleft. This removal occurs in 2 ways. 1. Acetylcholine diffuses away from the synapse. 2. Acetylcholine is broken down by the enzyme acetylcholinesterase to acetic acid and choline. Choline is then transported into the axon terminal for the resynthesis of acetylcholine.
So that always helps to kind of see it in motion like that. So I hope that helped in terms of going through the steps. But that's a critical aspect to understand how we get those muscle cells to contract. So you got to understand those steps, both from the neural side as well as the muscle side. Okay? Now, interestingly enough, when that's working, great, your muscles will contract. But we want to be able to get them to contract when we want to. We want to get them to relax when we want as well. And there are some types of diseases, disorders, drugs that interfere with that. So, for instance, myasthenia gravis is actually involving a shortage of those ACH receptors. And so this one is what we call an autoimmune disease. So autoimmune disease is where your own defenses are attacking your tissues. We'll also talk about, you talk about neurotoxins, like sometimes you hear about them in terms of venom and things like that, poison dart frogs, etc., right? You have native peoples that will put poison dart frog of the toxins on their arrows. And so same type of things can happen, is that it will basically affect this. Now, sometimes it keeps you from contracting your muscles, right? So it's a paralytic in that way. So it keeps you from contracting your muscles because it doesn't allow the communication to occur. In other cases, like if you're talking about getting botulism, so if you're talking about bacteria that can give you botulism, dented cans and all that type of things, what that also affects the neuromuscular junction. But the lockjaw that you can get when you get botulism comes from, again, these toxins affecting the neuromuscular junction. But rather than paralyzing them and keeping them from working, it actually keeps them contracted instead. So it decreases the uptake and the recycling, like we saw, of that acetylcholine. Okay, so what we find then is that we've gotten to a place where we've gotten the signal to the muscle cell itself. The next step is to see how that signal spreads along the muscle cell. It's going to get deep inside. It's going to get down to where the actin and the myosin are, and the troponin and the tropomyosin, and we're going to see how they respond in order to do that sliding that we say is such a critical part of that sliding filament theory or mechanism. So that's on the horizon.